Okay, from today onwards, let's discuss about biochemical mechanism. It is also a part of 301. So that means organic reaction mechanism. So in this chapter, it is what is called as an improved version of vitamins. So in the vitamins, we are discussing only the biological importance. So, but here, so we are looking the biological importance of vitamins mechanistic role. So more particularly, we will look for only the mechanism or mechanistic pathway, how the small molecules are synthesized in biological system. So that's what we are discussing in this chapter. More particularly, we are discussing the mechanistic role of the following in living system. So thymine pyrophosphate, it's a coenzyme. Uh, for this coenzyme, the precursor is vitamin B1. It's also called as thymine. So that means vitamin B1, it's also called as thymine. In living system, it will convert as a coenzyme, the coenzyme called thymine pyrophosphate. Similarly, vitamin B6, it's called pyridoxine. In the living system, pyridoxine or pyridoxal is converted into pyridoxal phosphate, which is a coenzyme. Of course, I will tell you what is the role of coenzyme. So coenzymes are the required by the enzymes to catalyze reactions in living system. Right. So pyridoxal phosphate is obtained uh, from the uh, vitamin B6, pyridoxine. Coenzyme, so it is also a coenzyme A more particularly. It will act as a source of 2 carbon unit. It will uh, uh, acetylate the amino acids, hydroxy group and amino group and other things in living system. So lipoic acid, it's also a sulfur containing one, and it is what is called as it is used in living system. Uh, a specific role we will discuss in uh, in this chapter. So biotin, it's a vitamin uh, B3. This biotin, it's a vitamin, but in the uh, living system, biotin is converted as a biotin decarboxylase. So it will act as a decarboxylating agent in the living system. And folic acid, it's a vitamin M. Okay, it is required for the dehydrogenating agent in the living system. So vitamin KH2, so more particularly we are aware about phylloquinone and hydroquinone. How the phylloquinone is converted into hydroquinone, that we are discussing under the heading of vitamin KH2. More particularly naphthoquinone and hydronaphtha. Flavin adenine coenzymes. So I think you are aware about FMN, flavin mononucleotide, FAD, flavin adenine dinucleotide. So these two are the living oxidizing reducing agents. So their role in oxidizing reducing agent in living system. Niacin, nicotinamide containing vitamin is called niacin, and its role, how the alcohol is converted into acetaldehyde, okay, by employing dehydration. So that we are discussing here. So coenzyme B12, I think you are aware that this is coenzyme B12 is responsible for anemic patients, and its role in the living system we will discuss in this chapter. So these are all the coenzymes we are discussing under the heading called biochemical mechanism. So what I will do, I will take one by one, and I will explain the reaction as well as the mechanism. So I will go to the first one, what is called as thymine pyrophosphate. So thymine pyrophosphate. So you know that up to this hydroxy group, okay, oxygen, it's called thymine. It's also called as vitamin B1. If this hydroxy group is phosphorylated in the living system, okay, that means two phosphorylated. If it is a one phosphorylated, then it's called thymine phosphate. If it is a two phosphorylated, two phosphorus groups are joined to vitamin B1, then it's called thymine pyrophosphate. And one more thing, you can observe that all the things, okay, molecules, more particularly amino acids or the coenzymes in the living system, they will exist in the form of ions, okay, that may be cation or anion. There won't be free groups. And here also, this NH2 won't be in free condition, because it will react with the acid part of the enzyme, and there will be a bond formation with the protein. Otherwise, what I can say that carboxylic end of the protein can react with this amine, and it will form a bond. Okay, that's what I can say. So we are what more particularly we are discussing about the thymine pyrophosphate. The role of TPP in TPP means thymine pyrophosphate in non-oxidative decarboxylation. Oxidative decarboxylation, non-oxidative decarboxylation. Oxidative decarboxylation means where there is an involvement of oxidizing and reducing agent. More particularly, not about the what you are aware about the oxidizing agents such as KMnO4, potassium dichromate, and all those things, but not those things. Those are the synthetic one. But in living system, FAD, FMN, niacin, okay. So these are the living oxidizing reducing agent. So if these are involved in the decarboxylation of alpha keto acids, so then it's called oxidative decarboxylation. If the living oxidizing and reducing agents are not involved, only the TPP only involved in the decarboxylation of alpha keto acids, then it's called role of TPP in non-oxidative decarboxylation of alpha keto acids. Suppose if there is a involvement of living oxidizing agents such as FAD, FMN, niacin, then such decarboxylation is called oxidative decarboxylation of alpha keto acids. So first thing you should know what is alpha keto acid. So, I think you are aware about the carbohydrate cycle. So, in the carbohydrate cycle, pyruvate or pyruvic acid is the end product. So pyruvic acid belongs to a keto acid. Why? Because a ketone and carboxylic acids are present in pyruvic acid. So that means if pyruvic acid undergoes decarboxylation only with the help of TPP, then it's called role of non-oxidative decarboxylation. Suppose a pyruvic acid undergoes decarboxylation with the help of living oxidizing agents, then it's called oxidative decarboxylation. Similarly, if the same pyruvic acid is used in the formation of acetol, okay, then it's called role of TPP in the formation of acetols. So these are the three types of reactions we are discussing under the heading of thymine pyrophosphate. So for thymine pyrophosphate, the precursor is thymine. Thymine is a vitamin B1. It is nothing but a vitamin B1. So that means if this oxygen, if there is no two phosphorus moiety, if this hydroxygen, if it form a bond with hydrogen, then it's called vitamin B1. And right. So then what is the source of vitamin B1? Hulls of rice kernel is the source of vitamin B1. Then what is the deficiency effects? It causes beriberi. Deficiency of vitamin B1 causes beriberi. Okay. So what I will do, with this, I will go for a next slide. In the next slide, I am taking the role of TPP in non-oxidative decarboxylation. Role of TPP in non-oxidative decarboxylation of keto acids. So this heading I am taking, and I will discuss the reaction first and then I will go for a mechanism. So I will go to the next slide. So before going to the reaction and mechanism of non-oxidative thing, so first I will explain how the active site is formed in TPP. So this is a TPP, and here I am mentioning enzyme part. So enzyme part of the base. So otherwise base part of the enzyme can obstruct this proton. So then what are the base part of the enzyme? I think you are aware that enzymes are nothing but a proteins. So then what are proteins? Proteins are nothing but a, for that proteins, the monomers are amino acids. So you know that amino acids means amino acids are of acidic amino acid, basic amino acids, neutral amino acids. So the base means lysine. Lysine means CH3 thrice NH2. The side chain is NH2 is contained. That can obstruct this proton. That means base part of the enzyme can obstruct this proton. If the base part of the enzyme obstruct this proton means this bonded pair will sit on this carbon as a anion. So this anion and this positive charge. So this is the active species or otherwise active site of the thymine pyrophosphate. Okay. So this is how a active site is formed in TPP. So that means base part of the enzyme can obstruct this proton. Then this bond pair will sit on this carbon as a anion. So this part is the active site of TPP. So this is how the active site is formed. Sometime they will ask you for 2 marks question, write the structure of active site of TPP. So then you should be able to write this particular one. So with this, I will go to the next slide. So there I am discussing how the role of TPP in non-oxidative decarboxylation. So first I will discuss the reaction. So the role of TPP in non-oxidative decarboxylation of keto acid. So this is the structure of pyruvic acid. How we will get this pyruvic acid? So this pyruvic acid is the end product of carbohydrate cycle. That means the glucose is converted into pyruvic acid. So this pyruvic acid, when it is subjected to TPP, TPP means thymine pyrophosphate. So thymine pyrophosphate is a coenzyme. Then what is the enzyme? Enzyme is the pyruvic acid decarboxylase. Then how I can say that TPP is a coenzyme which is required by the enzyme called pyruvate decarboxylase. That pyruvate decarboxylase catalyzes the reaction of pyruvic acid to acetaldehyde. So sometimes they will ask you, how do you convert a pyruvic acid into acetaldehyde? Explain with a mechanism. So what is the role of TPP in the conversion of pyruvic acid into acetaldehyde? So these are the questions you can expect. Otherwise, role of TPP in non-oxidative decarboxylation of alpha keto acid. So that's also a question, 5 marks question. You should be able to answer. So, but here one thing you need to remember, TPP, it's only a coenzyme. Then what is enzyme? Pyruvate decarboxylase is the enzyme. TPP is the coenzyme required by the enzyme that catalyzes the decarboxylation of pyruvic acid. So that is in terms of words. So what is happening here? Carbon dioxide is going out. H is forming a bond with this carbonyl carbon. So that is how acetaldehyde is obtained, starting with pyruvic acid. It's only the reaction. So I will explain in the next slide how this reaction mechanistically happens. Okay, that I will explain in the next slide. So look here. So this is a pyruvic acid. This pyruvic acid, the base part of the enzyme can obstruct this param, and this bond pair will sit on this oxygen. So that is how pyruvate is obtained. And this compound name is pyruvic acid. Pyruvic acid is converted into pyruvate. How? With the help of base part of the enzyme can obstruct the proton, forms a pyruvate. Once the pyruvate is formed, the TPP, active site of the TPP, that means this carbon is having anion. No, that anion attack the electron deficient carbon. This carbon is the electron deficient, but not this carbon. Okay, because this is the one is the ketonic nature, this is the one is the carboxylate nature. So because of this anion coming over here, this anion will sit on this oxygen. That means resonance of carboxylate ion, this carbon is electron rich compared to this carbon. So this anion will form a bond with this carbon, and this bond pair picks up a proton from the proton source of the enzyme. So that is how the intermediate is obtained. That means there will be a new bond formation between this carbon and this carbon. This double bond is converted into single bond. OH, oxygen, is forming a bond with proton and converted into this format. At this moment, this anion can come over here, and this bond pair will come over here, and this bond pair will convert as a lone pair of the nitrogen. So that's what I have written here. So that means in this, from here to here, carbon dioxide will go out as a gas. So now the lone pair present on the nitrogen can come over here, and this bond pair picks up a proton from the proton source of the enzyme. So that means this carbon is converted into CH. That's what I have written here. And this lone pair is coming over means nitrogen attains a positive charge. At this moment, the base part of the enzyme can obstruct this proton. The base part of the enzyme can obstruct this proton. The bond pair can come over here, and this bond pair will sit on this carbon. That means regeneration of TPP, and this part is converted as a acetaldehyde. This is about what is called as role of TPP in the conversion of pyruvate to acetaldehyde. Okay, now tell me, is there any doubt up to this? Hello? Hello?
Sir.
No. Okay. So I will go to the next slide. In the next slide, I am explaining the oxidative decarboxylation. Oxidative decarboxylation of pyruvate. That I am explaining in the next slide. So look here, role of TPP in oxidative decarboxylation of alpha keto acids. This is only the pyruvate is the alpha keto acid. This is an acid, and this position is called keto, alpha keto acid. Alpha keto acid is a general name, pyruvate is the particular name. So pyruvate, when it is treated with COASH, COASH means it's coenzyme A. So coenzyme A, it will act as a source of acetyl group, and in this, so this H is going out. Sulfur is forming a bond with electron deficient carbon. So here there is a sulfur is there, but so what you can write, a sulfur bond is there. Okay, of course, this name of this compound is also called as acetyl. This group is called acetyl coenzyme A. But here sulfur is there. That sulfur, it will form a bond with the electron deficient carbon. So that's what you need to remember. So what is happening? The acetyl coenzyme A picks up this acetyl group, converted into acetyl coenzyme A. And here there is a role of TPP in the decarboxylation process. TPP will have a role of decarboxylation. And lipoic acid is also having a role. FAD is also having a role. NAD plus niacin is also having a role in the conversion of pyruvate to acetyl coenzyme A with the liberation of carbon dioxide. This is only the reaction, but I am not writing the mechanism at this moment because it involves coenzyme A, lipoic acid, TPP, FAD, NAD+. Maybe you will come to know at the end of this chapter, so what is the role of coenzyme A, how it will react, how it will act as a source of acetyl group. So those things you will come to know. You know that TPP, it will act as a decarboxylating agent. Lipoic acid, it's a disulfide linkage compound. How it will act as a disulfide oxidizing form and reduced form more particularly pentothenic acid. You are aware about pentothenic acid. What is pentothenic? Vitamin B3 is called pentothenic acid. So pentothenic acid in the living system, it will act as a lipoic acid. That I will explain how the lipoic acid is involved in the living system. FAD means flavin adenosine dinucleotide. It will act as a dehydrogenating agent, okay, and NAD plus niacin, okay, it's an oxidized form. This NAD plus is the oxidized form, NADH is the reduced form. So NAD plus can accept a hydride from the substrate and convert it into NADH. That the role of NAD plus also I will explain. So in this process, pyruvate is converted into acetyl coenzyme A, and pyruvate also undergoes decarboxylation. So that's only the part I am explaining at this moment, and the remaining coenzymes role you will come to know within this chapter discussion. So this is the only the reaction part I am writing in the case of oxidative decarboxylation of alpha keto acid, because here there is a involvement of many coenzymes in the reaction. So because of that, the other coenzymes mechanistic part we will discuss. By that time, we will come to know how this pyruvate is converted into acetyl coenzyme A. So with this, I will take up one more heading, that is role of TPP in the formation of acetols, that I am taking in the next slide. So first I will mention the, this is the one, role of TPP in the formation of alpha acetol. So first you should know what is acetol. So here is CH3CO, CHOH, CH3 is called acetol. So this acetal, how it forms in living system, that we will discuss now. So as usual, you are aware that pyruvic acid, so pyruvic acid is the end product of carbohydrate cycle. So the base part of the enzyme can obstruct this proton, forms an anion, and the anion is called pyruvate. So to this pyruvate, the active site of thymine react. So that means anion part can react here, and C double bond O is converted into COH. Next step, it's a decarboxylation. This anion can come over here. This bond pair will form a bond with a double bond with this carbon, and this bond pair will convert as a lone pair. That's what I have written here. At this moment, again the lone pair is involved in bond formation. At this moment, this bond pair react with the one more equivalent of pyruvate. So one more equivalent. But suppose this bond pair react with proton, means it's nothing but a decarboxylation of pyruvate. Instead of reacting with proton, if it is reacting with a one more equivalent of pyruvate, then there is a formation of an intermediate like this. At this moment, the base part of the enzyme can obstruct this proton, and this bond pair can come over here, and this bond pair will sit as a anion. That means the regeneration of TPP is formed. So now you can observe that CH, CO, this one, CO, and CH, OH, and then CH3, CO2 minus. This is the one, is CO2 minus. At this moment, again, this moiety undergo decarboxylation. So how the decarboxylation? This bond pair is coming over here, This bond pair is coming over here. This bond pair picks up a proton. So that is how ene diol is obtained. This ene diol undergo enol ketone tautomerism. So that means this bond pair is coming over here. This bond pair picks up this proton, giving rise to acetol as the product. Okay. Now tell me, is there any doubt up to this? Any doubt? So I am discussed about the TPP, its role in non-oxidative decarboxylation of keto acids, oxidative decarboxylation of keto acids, and the role of TPP in the formation of alpha acetols. So that's all about the TPP. Now tell me, is there any doubt in TPP? TPP means thymine pyrophosphate. It is a coenzyme. Okay. How the coenzyme is formed? It is formed, and for this TPP, the precursor is vitamin B1. In other word, in second semester, in vitamins, what you learned that vitamin is a precursor required by the coenzyme called TPP. TPP is the coenzyme that is required by the enzyme that catalyzes the decarboxylation of keto acid. That's what you learned. But here you are learned about mechanistically how the keto acid is converted into undergoes decarboxylation and converted into acetaldehyde, and how the pyruvate is converted into acetal, and how the pyruvate is converted into acetyl coenzyme A. So these are the things we discussed under the heading called TPP. But here you have to remember that what I am taking the coenzyme part. So the coenzyme part is responsible for the changes of the substrate into a product, and this coenzyme is usually all the time it will be associated with the enzyme. Is it clear? Is there any doubt up to this?
No, sir.
Okay. So shall I go to the next? Shall I take one more coenzyme? So that means I have to go for a pyridoxal phosphate. So if I am taking the pyridoxal phosphate means it will take another 20, 25, 20 to 25 minutes or half an hour. Shall I go for one more pyridoxal phosphate, or you want me to take in the next class? Shall I complete the pyridoxal phosphate? Hello? Hello? Okay. What I will do? I will take pyridoxal phosphate. So next, with this, I will go to the next slide. So pyridoxal phosphate. So I think you are aware that pyridoxal phosphate is the coenzyme part of the vitamin B6. What is vitamin B6? Vitamin B6 is the pyridoxamide, pyridoxal, okay, uh, pyridoxine, okay. Three forms are called vitamin B6. So usually it's called pyridoxamine. It's called vitamin B6. So vitamin B6 in the living system, it will convert as a pyridoxal phosphate. So this is the pyridoxal phosphate. So here, if there is a CHO, then it is called pyridoxal. Here, if there is a CH2, NH2, then it's called pyridoxamine. If there is a OH, then it's called pyridoxine. And the CHO, it will form a bond with the enzyme part. Okay, E stands for enzyme. So amino part of the enzyme react with the CHO of the pyridoxamine and converted into a PLP. Similarly, OH is the if there is a OH, it's called if there is a OH, if there is a CHO here, then if there is a OH here, then it's called pyridoxine. If this O is phosphorylated, usually it will phosphorylate in the living system. Similarly, CHO, it will form a skip base with the enzyme part of the amino group. This is called enzyme bound PLP. Enzyme bound PLP means this is only. Otherwise, in living system, PLP is nothing but a structure. Okay, so what we are discussing, the role of PLP in the transamination. So you know that here enzyme bound amine is there. Suppose another amine, where you will get another amine? Maybe the amino acid, another amino acid. The amino acid may be from the protein source. So if the amine replaces this enzyme, then it's called transamination. I think you are aware about transesterification. What is transesterification? Methanol is replaced with ethanol. Ethanol is replaced with isopropanol. So it's called transesterification. So here also, if this amine is enzyme bound amine is replaced with another amino acid, then it's called transamination. Decarboxylation, role of PLP in decarboxylation. You know that amino acid is there. In that amino acid, if carboxylic acid is going out as a decarboxylation, then it's called decarboxylation. So that means in the living system, decarboxylation occurs, okay, with the help of PLP. And one more thing, decarboxylation of the alpha keto acid occurs through TPP. Okay. Then role of PLP in the elimination reactions of amino acids. So what are eliminate? I think you are aware that what is called as serine. So what is serine? Serine is a amino acid. So more particularly, hydroxy group containing amino acid. This hydroxy group, is it possible to eliminate? Okay, so then such type of reactions are called elimination reactions. So elimination reactions are catalyzed by PLP. What is PLP? PLP is a coenzyme. So coenzyme form of vitamin B6. Then coenzyme PLP is required by the enzyme that catalyzes the elimination reactions. PLP is the coenzyme required by the enzyme that catalyzes the dealdolization. That means here aldehyde is there, no? Aldehyde can be removed, okay, with the PLP catalyzed enzyme reactions. And similarly, decarboxylation, PLP catalyzed. So otherwise what we can say that PLP is a coenzyme required by the enzyme that catalyzes the decarboxylation of a substrate. Okay, PLP is the coenzyme required by the enzyme that enzyme catalyzes the reactions of transamination. Okay. Similarly, PLP is the coenzyme that required by the enzyme that catalyzes the racemization of reactions of amino acid. So enzyme invertase, gymase. So these are the enzyme will act as a catalysis of racemization. Then PLP is the uh, coenzyme required by the enzyme that catalyzes the amino acid metabolism in the living system. So for all these reaction, PLP is the coenzyme. For the PLP is formed via by the conversion of vitamin B6. So vitamin B6 is the pyridoxine. Vitamin B6 in living system, it will convert as a PLP. Is it clear? Is there any doubt about the role of PLP in living system? Any doubt? Is there any doubt up to this?
No, sir.
Okay. So I will take up one by one. So in the next, I am taking transamination, role of PLP in transamination. So directly I am going for mechanism. So this is the amino group, and this is the enzyme bound PLP. So you know that nitrogen attains a positive charge. Since there is a positive charge on this nitrogen, this bond pair try to convert as a lone pair of this nitrogen. So in such cases, the lone pair present on this amino acid, it will form a bond with this carbon. And one more thing you can observe, this O minus, it will form a hydrogen bond with this proton of the uh, uh, amino group of the enzyme board. So once there is a new bond formation between the amino group of the amino acid to this carbon, then what will happen? This complete amino group is coming over here, and this amino group of the amino acid, it will form a bond with this carbon. So you can observe the color mapping. So this pink color is from the amino acid one, and this black color is the amino acid bound PLP one. So that is here NH enzyme and NH enzyme. Why NH enzyme? Because this bond pair is converting as a lone pair. So that is the reason here the lone pair is present on this nitrogen. At this moment, you know that nitrogen is a positive charge. Since there is a positive charge, this bond pair can convert as a lone pair of this nitrogen. Otherwise, what it will do, you know that this lone pair present on the nitrogen can form a bond with the proton. Otherwise, this bond pair itself can form a bond with the proton source of the enzyme. So in such cases, this bond pair will come over here. So that means it will form a double bond between the nitrogen of the amino acid and carbon of the PLP. So that means that is how a double bond is formed. So here the living group. What is living group? Enzyme bound amine is the living group. So what is happened now? So the amino group of the enzyme is going out, and amino group of the amino acid, it will form a bond with this carbon. So this is only, it's called transaminated PLP. Okay, a amino acid group is form a bond with PLP. So in this case, enzyme bound PLP. This is called enzyme bound PLP, and this is amino acid bound PLP. So what it will do in future, this undergoes this part undergoes decarboxylation. Up to this is called transamination. Okay, is there any doubt in transamination? Because in next slide I am taking a decarboxylation. So that is the transamination means only this much. The PLP, enzyme bound PLP, when it is react with the amino acid, okay, this enzyme bound amino group is going out, and amino acid, it will form a bond with the PLP1. So that is the transaminated PLP. So this is only the role of PLP in transamination. In the next slide, I am taking the decarboxylation. So for the decarboxylation, I will start with this mighty transaminated PLP itself in the next slide. So I will go to the next slide. So look here, this is the amino acid transaminated PLP. How the decarboxylation occurs? The anion CO minus is there, no? This anion can come over here, and this bond pair will come over here, and this bond pair will come over here, this bond pair will come over here, this bond pair will convert as a lone pair of this nitrogen. That is how intermediate is formed. But here there is a loss of carbon dioxide. Since there is a loss of carbon dioxide, it's called decarboxylation. So then what we can say that? So enzyme bound, what you call amino acid bound PLP undergo decarboxylation. So here, so one enzyme should be there. So what is that enzyme? Decarboxylase enzyme. Okay, decarboxylase is the enzyme, okay, which responsible for the decarboxylation of the enzyme bound PLP, means amino acid bound PLP. Suppose if R is equal to hydrogen, then what is the enzyme responsible for the conversion of decarboxylation? If R is equal to H, then we can say that glycine. Okay, glycine decarboxylase is the enzyme that catalyzes the decarboxylation of the glycine bound PLP. Okay, that's what you need to remember. Then what will happen? Again, the lone pair can form a bond with this, can form a double bond here. This pi bond will move over here. This pi bond will move over here. This pi bond picks up a proton from the proton source of the enzyme. So that is how an intermediate is obtained. At this moment, hydrolysis. Hydrolysis means CH double bond N. So that means here there is a breakage. So breakage in the sense this carbon is converted into CHO, and this nitrogen is converted into NH2. So that is how a decarboxylated compound is obtained. Then what is the role? I think you are aware about the histidine. Histidine is a amino acid. So histidine in the living system undergoes decarboxylation converted into histamine. Histamine is a very important protein or intermediate which is formed in living system, and that is very much required for cold to reduce the cold and cough, to get a immune system, and it is also very much required for the skin. So that's what maybe you can look into the role of histamine in the living, role of histamine, that you can look into that. You will get a lot of information. So histamine is the decarboxylated product of histidine. Histidine is a amino acid, right? So this is about what is called decarboxylation. So explain the role of PLP in decarboxylation means you have to write all these things. Okay. Is there any doubt in decarboxylation? If there is no doubt in decarboxylation, I will go for a next one. So next may be the elimination reactions. I will go to the next slide. So here dealdolization, role of PLP in dealdolization. Okay, removal of removal of how it takes place compounds, and this dealdolization takes place compounds having beta hydroxy group. So this is a carboxylic acid. This position is called alpha. This position is called beta hydroxy group. For example, serine. Name of this amino acid is serine. And you know that the first step, transamination. So this PLP should react with amino group of the serine. What you will get? This amino group will form a bond over here. Then it's called trans-serine PLP. That serine PLP react with the amino acid decarboxylase. Amino acid decarboxylase means, otherwise what we can say that instead of amino acid we can say that serine decarboxylase is a enzyme that catalyzes the reaction of serine to the glycine. So glycine is the product. Here, what is happening? Dealdolization. So it's not decarboxylase, it's a dealdolase. It's not decarboxylase, it is a dealdolase, LASE. It's an enzyme. So serine dealdolase is the enzyme which is responsible for the conversion of serine to the glycine in presence of PLP. Is the words explanation about the dealdolization. Okay, is it clear? So this is what is called as a reaction. In the next slide, I will take a mechanism. So here a glycine part is forming a bond with the PLP. And in this reaction, magnesium is also having a role. Why? Because this magnesium, it will form a bond with the oxygen of the carboxylate of the serine and also oxygen of the phosphate 1. So that means magnesium, it will bind the 2 oxygen molecule, 1 oxygen from the serine, another oxygen from the phosphate. Right, this is the key intermediate. And here OH, okay, the base part of the enzyme can obstruct this proton. And here I am showing how the dealdolization occurs. So that means OH, okay, proton, okay, this proton is captured by the base part of the enzyme. In such cases, the bond pair can come over here. The bond between this carbon and this carbon breaks. So that is how dealdolization occurs. So that means H HCOH. So this part is going out as a aldehyde. And this bond pair will form a bond with double bond with the nitrogen. This bond pair can come over here. This bond pair come over here. This bond pair will convert as a lone pair of the nitrogen. So that is how an intermediate is formed. Again, the lone pair can come over here. Bond pair will go over here. This bond pair will form a double bond here. This bond pair picks up a proton from the proton source of the enzyme. Then you will get an intermediate like this. So this intermediate, again hydrolysis. Here there is a hydrolysis giving rise to glycine. So this is how the serine is converted into glycine. So what is the role of serine decarboxylase? Serine decarboxylase is a enzyme. That enzyme requires a PLP bound serine. That enzyme require a PLP. So what is the role of PLP? PLP it will bound the serine. Serine means substrate and catalyzes the conversion of dealdolization and converting a serine into glycine. So I will repeat what is called as serine decarboxylase is a enzyme which is converts serine into glycine. How it will convert? By the dealdolization. So in this process, PLP is a coenzyme. And in this process, magnesium, it is having a role. It will bind the two oxygen, one oxygen from the serine one, another oxygen from the phosphate one. It will form a bond with the magnesium 2 plus. Is it clear? Is there any doubt how this is a dealdolization? And this dealdolization is require a coenzyme called PLP. And PLP is bound to the enzyme called serine decarboxylase. So with this, I will go for the next one. It's called elimination reaction. So elimination means a small molecule should go out. So that I am explaining in the next slide. So role of PLP in the elimination reactions. So here again I am taking a serine. So serine, when it is treated with the PLP bound enzyme, PLP bound enzyme that catalyzes the conversion of serine to alanine by elimination. So here water is the product which is eliminated during the reaction. So this is a reaction. So serine is converted into alanine. It's a elimination reaction. Serine, if it is converted into glycine, then it is called dealdolization. Okay, I will go for a mechanism. So same thing I am taking means what is called a serine bound PLP. But at this moment, the enzyme, the base part of the enzyme does not capture a proton of hydroxy, but it capture a alpha proton. Okay, alpha proton means yes, alpha proton. Base part of the enzyme capture a alpha proton. Then what will happen? The anion means bond pair will come over here. This bond pair come over here. This bond pair come over here. This bond pair convert as a lone pair. Again, this lone pair can come over here, this bond pair will come over here, this bond pair will come over here. This bond pair form a double bond with this carbon, and hydroxy will go out along with the proton, and that proton takes from the proton source of the enzyme. Water is going out. Say intermediate is formed like this. So this on hydrolysis, skip base hydrolysis, CH double bond NH is called skip base. This hydrolysis NH2 is obtained, and later on this double bond is reduced in the living system and converted into here CH3. So here I am not written a CH3. You need to write down. This is not glycine, it is a alanine. So here you have to write the CH3. Okay. So this this that's all about the PLP. The role of PLP in transamination, role of PLP in decarboxylation, role of PLP in dealdolization, role of PLP in elimination reaction. Sometimes they will ask you the 5 marks question, explain the role of pyridoxal phosphate. Otherwise, write a note on pyridoxal phosphate in biochemical mechanisms. So today we discussed two components. One is TPP, another one is PLP. Role of TPP and PLP in living system. Okay, tell me, is there any doubt up to this?